Good day, Doc, and everyone. I am Angirkani. Our teammates are Aarti, Aishwarya, Ajay Kumar, Angirkani, Anusha, Anushuptika, Ashika Pitti, Delsa, Devilita, and Dilsa Taran. Our histology topic is oral cavity, lips, teeth, tongue, and salivary gland.
Hello everyone, let me introduce myself. This is Aishwarya. As you can see on the screen, today my topic is histology of oral cavity. The oral cavity is lined with stratified squamous epithelium which may be keratinized, partially keratinized or non-keratinized depending on the location. The keratinized cell layers resist damage from abrasion and are best developed in the masticatory mucosa on the gingiva, gum and hard palate. Throughout the oral cavity, the epithelium contains transient antigen presenting cells and rich sensory innervations. Non-keratinized squamous epithelium predominates in the lining mucosa over the soft palate, cheeks, the floor of the mouth, and the oropharynx, the posterior region of the oral cavity, leading to the esophagus. Medical application. Viral infection with herpes simplex 1 cause death of infected epithelial cells that can lead to vesicular or ulcerating lesions of the oral mucosa or skin near the mouth. In the oral cavity, such areas are called canker sores, and on the skin they are usually called cold sores or fever blisters.
Hello everyone, I am Angirkani. Our next topic is lips. The well-developed core of stained muscles in the lips or labia makes these structures highly mobile for injection and speech and other forms of communications. Both lips have a three differently cored surface. The internal mucous surface has a lining mucus with a thick non-considered epithelium and many minor labia salivary glands. The red vermilion zone of each lip is covered by very thin cartilage stratified squamous epithelium and is anal between the oral mucus and skin. This region lacks salivary or sweat glands and it keeps moist with a saliva from the tongue. The underlying connective disease is very rich in both sensory intervention and capillaries, which impact the pink color to these regions. The outer surface has a thin skin connecting of epidermal and terminal layers, sweat glands, and many hair follicles with a sebaceous gland.
Hello everyone, I am Anushrutika. Today I am going to talk about teeth. Teeth. In adult human, there are normally 32 permanent teeth, arranged in two bilaterally symmetric arches in the maxillary and mandibular bones. Each quadrant has 8 teeth, 2 incisors, 1 canine, 2 premolars, and 3 permanent molars. 20 of the permanent teeth are preceded by primer teeth, deciduous or milk teeth, that are shed. The others are permanent molars with no deciduous precursors. Each tooth has a crown exposed above the gingiva and constricted neck at the gum, and one or more roots that firmly into bone sockets in the jaw called dental alveoli. The crown is covered by very hard acellular enamel and the roots by a bone-like tissue called cementum. These two covering meet at the neck of the tooth. The bulk of a tooth is composed of another calcified material, dentin, which surrounds an internal pulp cavity. Dental pulp is highly vascular and well innervated and consists largely of loose mesenchymal connective tissue with much ground substance, thin collagen fibers, fibroblasts, and mesenchymal stem cells. The pulp cavity narrows in each root as a root canal which extends to an opening apical foramen at the tip of each root for the blood vessels, lymphatics, and nerves of the pulp cavity. The periodontal ligaments are fibrous connective tissue, bundles of collagen fibers inserted into both the cementum and the alveolar bone. Dentin. Dentin is a calcified tissue harder than bone, consisting of 70% hydroxyapatite. The organic matrix contains type 1 collagen and proteoglycans secreted from the apical ends of odontoblasts. Taller polarized cells derived from the cranial neural crest that line the tooth pulp cavity. Mineralization of the predentine matrix secreted by odontoblast involves matrix vesicles in a process similar to that occurring in osteoid during bone formation. Enamel. Enamel is the hardest component of the human body, consists of 96% calcium hydroxyapatite and only 2 to 3% organic material, including very few proteins and no collagen. Enamel consists of uniform interlocking columns called enamel rods or prisms, each about 5 mm in diameter and surrounded by a thin layer of other enamel. In a developing tooth bud, the matrix of the enamel rods is secreted by tall polarized cells the amyoblast, which are part of a specialized epithelium in the tooth bed called the enamel organ. Growth of the hydroxyapatite crystals to produce each elongating enamel rod is guided by a small 20 kDa protein amylogenin, the main structure protein of the developing enamel. Periodontium. The periodontium comprises the structure responsible for maintaining the teeth in the maxillary and mandibular bones and includes the cementum. The periodontal ligament and the alveolar bone with associated gingiva cementum covers the dentine of the root and resembles bone but is avascular. It is thickest around the root tip where cementocytes resides in lacunae with the process in canaliculi, especially near the cementum surface. The periodontal ligament is fibrous connective tissue with bundled collagen fibers, sharpie fibers binding the cementum and the alveolar bone. The alveolar bone lacks the typical lamellar pattern of adult bone but has osteoblasts and osteocytes engaging in continuous remodeling of the bone matrix. Around the periodontium, the keratinized oral mucosa of the gingiva is firmly bound to the periosteum of the mandibular and maxillary bones. Between the enamel and the gingival epithelium is the gingival sulcus, a group up to 3 millimeter deep surrounding the neck.
Hi everyone, I am Devlalita, histological view of tongue. The tongue is a mass of striated muscle covered by mucosa which manipulates ingested material during mastification and swallowing. The muscle fiber are orientated in directions allowing high level of mobility. The lower surface of tongue is smooth with typical lining mucosa. The dorsal surface is irregular, having hundreds of small protruding papillae of various types of anterior two-thirds and merged lingual tunnels posterior third of the root of the tongue. The papillary and tonsillar areas of the lingual surface are separated V-shaped groove called the sulcus terminals. The lingual papillae are elevations of the mucous membrane that assumes various forms and functions. There are four types of papillae: filiform papillae, fungiform papillae, foliate papillae, valate papillae. First, talk about filiform papillae. Filiform papillae are very numerous, have an elongated conical shape and heavily keratinized, which gives the surface or grey or whitish appearance. They provide a rough surface that facilitates movement of during chewing. Let's talk about fungiform papillae. Fungiform papillae are much less numerous, lightly keratinized and interspread among the filiform papillae. They are mushroom-shaped, well vascularized and innervated course of lamina propria. Let's talk about foliate papillae. Foliate papillae consist of several parallel tracts each side of the tongue anterior to the sulcus terminals but are rudimentary in humans and older individuals. Valid papillae. Valid papillae, another name of circumflex papillae, are the largest papillae. Its diameter to 1 to 3 millimeter. 8 to 12 valid papillae are normally aligned just in front of terminal sulcus. Duct of several small serous salivary glands empty into the deep moat-like groove surrounding each papillate. Taste buds are ovoid structure. It is stratified epithelium to the tongue surface which sample the general chemical composition of ingested material. Approximately 250 taste buds are present lateral surface each valid papillae. It is present in fungiform and foliate but not characterized filiform and papillae. Taste bud has 50 to 100 cells, about half of the elongated gustatory cells, which turn the 7 to 10 day lifespan. At the apical end of the gustatory cells, microvilli project to muum, wide opening in the structure called taste pore.
Hello everyone, I'm Ashika. Uh, Now we'll see about the histology of the salivary glands. Exocrine glands in the mouth produce saliva which has digestive, lubricating and protective functions with a normal pH of 6.5 to 6.9. Saliva also has an important buffering function and in some species it is also important for evaporative cooling. Saliva is modified by the cells of the duct system, draining the secretory units with much sodium and chloride reabsorbed while certain growth factors and digestive enzymes are added. The IgA antibody complex released into the saliva provides defense against specific pathogens in the oral cavity. Parasympathetic stimulation, usually elicited through the smell or taste of food, provokes a copious watery secretion with relatively little organic content. Sympathetic stimulation inhibits such secretion and produces the potential for dry mouth, often associated with anxiety. There are three epithelial cell types comprise the salivary secretory units. First, it's the serous cells. They are polarized protein secreting cells, usually pyramidal in shape, with round nuclei, well stained rough endoplasmic reticulum, and apical secretory granules. Joined apically by tight and adherent junctions, serous cells form a somewhat spherical unit called an acinus, with a very small central lumen. Serous acinar cells secrete enzymes and other proteins. Next, it is mucous cells. They are somewhat more columnar in shape with more compressed basal nuclei. Mucous cells contain apical granules with hydrophilic mucins that provide lubricating properties in saliva but cause poor cell staining in routine preparations. Mucous cells are most often organized as cylindrical tubules rather than acini. Mixed salivary glands have tubuloacinous secretory units with both serous and mucous secretion. The next is myoepithelial cells. They are found inside the basal lamina surrounding acini, tubules and the proximal ends of the duct system. These small flattened cells extend several contractile processes around the associated secretory unit or duct, and their activity is important for moving secretory products into and throughout the ducts. The parotid glands located in each cheek near the ear are branched acinar glands with exclusively serous acini. Serous cells of parotid glands secrete abundant alpha amylase that initiates hydrolysis of carbohydrates and proline-rich proteins with antimicrobial and other protective properties. The submandibular glands, which produce two-thirds of all the saliva, are branched tubuloacinar glands, having primarily serous acini, but with many mixed tubuloacinar secretory units. Serous cells of these mixed units are mostly in peripheral crescent-shaped groups called serous demilunes, aggregates formed at least in part artifactually when the adjacent mucous cells swell during routine tissue preparation. Basolateral membrane enfoldings of the serous cells facilitate and water electrolyte and water transport. In addition to alpha amylase and proline rich proteins, serous cells of the submandibular gland secrete other enzymes including lysozyme for bacterial wall hydrolysis. Next, it is the sublingual glands. The smallest of the major glands are also considered branched tubuloacinar glands. But here, secretory tubules of mucous cells predominate and the main product of the gland is mucus. The few serous cells present in demilunes and amylase and lysozyme to the secretion. So these are the pictures for the parotid gland, the submandibular gland and the sublingual gland. Thank you.